I want to thank the organizers for the opportunity to present this exciting collaboration um, with PacBio and on multiplex long read sequencing of ataxia repeat expansions. And just to start off, I want to acknowledge my collaborators on this project, specifically those from PacBio, Yuchi, Ginny, Janet, and John, who developed the panel that I will discuss, as well as my collaborator at the Mayo Clinic, Mark Van Blitterswyk. This has been an exciting collaboration that started not too long ago when we have some great data that I'm excited to share with everyone. So just to give a brief overview of ataxia, ataxia is a neurological symptom characterized by the incoordination of voluntary muscle movements. More broadly, it's used as an umbrella term to describe a group of degenerative diseases that affect movement and coordination. Moreover, these ataxic disorders typically result from lesions in the cerebellum. However, lesions in the peripheral nerve connecting the cerebellum as well as the spinal cord can also produce ataxia. The etiology of ataxia really falls under three main categories: acquired ataxias that result from an underlying issue, such as a tumor, sporadic ataxia, and lastly, hereditary ataxias, which will really be the focus of this talk. The hereditary ataxias, of course, are inherited, and therefore there's usually always an underlying genetic cause of these ataxias. The hereditary ataxias are a clinically and genetically heterogeneous group of inherited ataxic disorders, and they fall under a few different categories: autosomal dominant, which are usually referred to as the spinal cerebellar ataxias, the SCAs or SCAs, recessive ataxia mutations such as Friedreich's, X-linked, and mitochondrial. And what's unique about these ataxias, especially the spinal cerebellar ataxias, is there's distinct neuropathological and molecular signatures associated with these hereditary ataxias. A number of these ataxias have ubiquitin positive inclusions. Some of them have distinct RNA foci, as well as recent reports describing the presence of these abnormal peptide species referred to as dipeptide repeats. And so if you look at these spinal cerebellar ataxias, there are a number of them, and currently there are 48 different SCAs. And while a majority of these ataxias are caused by point mutations in the genome, a significant number of these are actually caused by expanded repetitive sequences. Also, just to note that Friedreich's, although a recessive mutation, is also caused by an expanded repeat sequence. And you can see here that these repeat sequences, though they lead to cerebellar ataxia, are slightly different in these various ataxias, though some are overlapping. Moreover, the location of these repeat expansions are within different genes, and even within these genes are within different loci. And so, this poses a unique problem for trying to screen and determine whether a patient might be harboring one of these expanded repeat mutations. Typically, ataxia repeat screening methods rely on traditional PCR-based methods. Normally, these are single tube assays. Moreover, they're limited in the ability to size the expanded repeats. And lastly, there's a bias in the amplification of expanded alleles. Moreover, they're kind of notoriously difficult to design multiplex screening methods to detect expanded repeat sequences, especially those that have high GC content and or are longer repetitive sequences. The multiplex systems that are available really only target the CAG ataxias. Moreover, they only target a limited number of the CAG ataxias, 4 or 5, and they're limited to a single individual per test. And so with PacBio technology, we can circumvent some of these issues surrounding the limitations associated with analysis of long repetitive sequences and repeat expansions underlying the spinal cerebellar ataxias. And so here is a diagram of the general workflow of the PacBio sequencing to target repeat expansion sequences. And key to this is the ability to use the CRISPR-Cas9 system to excise specific target loci. And this can be done with a pair of guide RNAs flanking this target region. And as long as this target region is accessible to the CRISPR-Cas9 nuclease, you can really design a unlimited hypothetical number of regions to access, target, and sequence. And the ability to multiplex this and barcode these libraries further allows high throughput analysis of multiple samples. And so this is the idea that Yuchi and the collaborators at PacBio had when they developed this no-amp SCA panel. And they designed a number of CRISPR-Cas guide RNAs targeting various loci known to harbor repeat expansions underlying various ataxias. And here's a table outlining the various ataxias that this panel can actually target. And so Yuchi had previously shown that that it was possible to use this system to sequence these ataxia or these genes harboring potential ataxia repeats, and we extended this this to show that this can be done on actual patient samples. And so combining this panel, this novel panel, with the ability to sequence through long repeats, we really provide us a unique opportunity to sequence these expansions. And so in this initial pilot experiment that we recently performed, we took 15 different spinal cerebellar ataxia samples, and we collected DNA from these samples using the Circulomics nanobind kit. The DNA collected from this was DNA from the cerebellar cortex, and the patients in this cohort were known to have various ataxia expansions previously diagnosed, either molecularly or clinically. And in this initial experiment, we had SCA1, SCA2, 3, 6, Friedrichs, and Doppler. And for the sequencing, we set up a few different paradigms. One, we did a multiplex experiment where we analyzed all samples with 1 microgram of genomic DNA. And then we kind of split this up across 3 cells and analyzed 5 samples per cell at 4 micrograms of genomic DNA. And I will discuss a little bit about this in a few slides. And so it was exciting to see, once we got the data back, that there was significant sequencing coverage for all the loci tested in this panel, as well as almost all of the samples. In fact, really only 2 samples had lower sequencing coverage, and this was likely due to poor DNA quality. However, you can see that the majority of loci had robust reads and coverage, and this allowed us to really be able to analyze these regions. And so just to go over this, just to go over one case in particular that had good sequence coverage and a majority of the reads mapped on target, is this sample 7. And this was a known SCA3 patient sample. And you can see here on the left-hand side, is a waterfall plot depicting the sequencing reads, or the sequences that we got back from this panel. And notably, in this waterfall plot, where we plot each read on the y-axis, and the x-axis is the size of the read, you can see two distinct populations, and pretty much, split in half. And what you'll notice is these reads, each line represents a different read, are colored. And the blue represents CAGs, which are the known repeat expansions, or the known repetitive sequence in SCA3. And the other colors represent interruptions and or different sequences within each read. And when this data is plotted as a distribution plot, you can see that there really is two distinct distributions of repeat sizes. And you can attribute this to the wild type allele as well as to the expanded allele. And what you'll notice is in the wild type allele, it's pretty consistent. Most of the reads represent 20 CAG repeats, whereas on the expanded allele, you see a kind of broader distribution of sizes. However, the median expanded allele size is 69 repeats, which is significantly higher than the pathogenic cutoff of 45 repeats. And so this, this experiment really demonstrated that we can robustly detect expanded repeats in the ataxin-3 loci, which cause SCA3. And so, as I mentioned, we did two different amounts of DNA when we did these experiments, 1 microgram DNA input or a 4 microgram DNA input, to really see if this offered any novel insight. And what's nice about this is both the 1 microgram DNA input as well as the 4 microgram DNA input experiments demonstrate that the wild type allele has 20 repeats and the expanded allele 69 repeats. So they're consistent. The only real difference is that in the 4 microgram DNA input is that we see more CCS reads, which we would expect. And this is important for considerations regarding the multiplex or the high throughput nature of this assay. What is the lower limit of DNA needed in order to be able to accurately call the repeat sizes? And as I mentioned at the beginning of the talk, we sequenced a number of patients simultaneously in a high throughput manner. And the data presented here represent a number of different repeat expansions detected by the NOAMP ataxia panel. And you can see that we robustly detected repeat expansions in a number of loci underlying various ataxia disorders. And all of the sizing of the expanded allele in these disorders demonstrate that they're above the pathogenic cutoff for these disorders. And one thing to note too is that in some of these, for instance, in the SCA1 and SCA2, you can see distinct polymorphisms in the two alleles. So in the non-expanded alleles in the SCA1, for instance, there's two clear CAT polymorphisms, whereas there's two distinct CAAs in the SCA2, further demonstrating the dichotomy between the expanded and wild type allele. And one thing that's interesting is while we thought the majority of these that we knew the majority of the repeat expansions in these 15 patients, there were two actually that were clinically diagnosed with a particular spinal cerebellar ataxia, but upon analysis using this NOAMP ataxia panel, we saw that in fact they didn't meet the threshold for or the pathogenic cutoff for an expanded repeat. And checking the other repeat loci in this panel, it didn't meet that cutoff as well either. So, these two samples remain molecularly undiagnosed, but I think this this panel, further confirmed that it can, accurately detect when there's an expansion or not. And so, one thing unique about, for instance, the top case, the SCA3 case, this is from quite a long time ago. It's from 1995, and it was diagnosed clinically, and so there's clearly a discordance between the genetic and clinical diagnosis, and I think application of this ataxia panel will better inform the clinical diagnoses, because of course these won't always be 100% accurate with regards to the genetic diagnosis. And so, lastly, I just want to mention that this ataxia panel isn't only capable of detecting and sizing repeats, but there's a lot of literature in the ataxia field about flanking polymorphisms that either modulate or affect the aggregation of some of these peptides that are generated from these expanded repeats. And for instance, a recent paper demonstrated that a number of Ataxin-3, SCA3 cases have a unique polymorphism associated with the expanded allele. And so we went back to the data generated from the NOAMP panel, and we saw that most of or almost actually all of the SCA3 cases, you can see that these patients have this polymorphism, this TAA polymorphism. And so this is valuable data that, could possibly inform future therapeutic strategies, or provide researchers, a better understanding of potential or molecular outcomes that could result of this expansion. And so, it's just further highlighting the capabilities of this panel to not only capture the repeat expansion, but also important sequence information. And so, in summary, I just want to go over a few of the findings. We validated the NOAMP targeted sequencing ataxia panel to screen carriers harboring pathogenic repeat expansions. This was done in a high throughput manner, analyzing 15 ataxia patients at one time. We validated and sized pathogenic repeats in 13 of these patients harboring repeat expansions. And then we identified 2 potential ataxia cases that actually were discordant between the clinical diagnoses. And so obviously, this would allow us to then maybe use other techniques to determine if there is a genetic mutation in these patients, or if they lack a genetic variant. And lastly, it should be emphasized that the NOAMP targeting sequencing approach provides unparalleled benefits for targeting these repeat expansions. Most importantly is there's no PCR amplification bias. And you saw that where in the waterfall plots, where you got similar number of reads between the expanded and wild type allele. And this gives kind of, a little bit more faith in the ability to size both alleles. Moreover, this panel has, it is actually very adaptable because of the CRISPR-Cas9 targeting and the ability to barcode, these samples. You can really target any genomic locus and kind of make this panel modular. And maybe there's some genes that are rare that you want to take out, or maybe in the future a novel repeat expansion would be discovered underlying a neurological disorder, and you could hypothetically add that to this panel. And lastly, of course, inherent to the PacBio technology is the capability to sequence large repeats regardless of their GC content. And so this is important for not only these spinocerebellar ataxias, but other repeat expansion disorders, such as C9orf72. And so, it's really a great tool that I'm excited to use, and I feel fortunate to be part of this collaboration. And we're continuing this collaboration to better understand these repeat expansions. And so right now, in collaboration with Marka and Yuchi and everyone at PacBio, we're analyzing additional SCA cases. We want to try, obviously, more polyQ samples, other penta or hexamer repeat cases, as well as cases that are known to have be defined by pathogenic interruptions. Furthermore, we want to look at increased sample throughput. How many cases can we actually analyze at one time? We want to validate a number of these cases, validate their repeat size using more standard methods. Some of them we have sequencing information on, others we don't, and so it'll be nice to validate the ones that we can using more low throughput traditional methods. And functionally, we can correlate some of the repeat sizes and or interruptions that we detect using this technology with molecular and neuropathological signatures, which will possibly provide real insight into the pathogenesis of these neurological disorders. And just concluding, I want to again acknowledge the collaborators, Marka, Yuchi, Jenny, Janet, John. It's been a great collaboration. It's really just kind of started quickly, and we've been fortunate to get really, really great data. And again, this is such an awesome system that Yuchi developed there at PacBio, and so we're excited to continue moving forward with this collaboration. Thank you.